I would like to introduce Professor Kate Tatton-Brown. She is a medical genetics doctor working in UK, St. George's Hospital. She studied medicine at the University of Oxford before training in pediatrics and then in medical genetics. Kate, as part of a team of scientists and clinicians, first discovered TBRS in 2014. Since then, she's been working to characterize the clinical symptoms of TBRS and to develop clinical management guidelines. Some of this work has already been published, but work is ongoing. And she worked very hard to get here. Her flight was canceled. She had to run through airports and push people out of the way, and like a scene from a movie. So let's give her a really warm welcome, Dr. Kate Tatton-Brown.
Thank you very much, Jill. And what an amazing morning already. I mean, my mind has been blown by all the talk so far this morning. So it's a great, great privilege to be here with you all. There's a bit of a warning before I start talking today that those of you who were here yesterday, there's a bit of duplication in what I'm saying. And I really hope that helps to reinforce things and not bore you so that you fall asleep. But please feel free to fall asleep if you so wish. So my name's Kate Tatton-Brown, and as Jill said, I am a clinical geneticist, or you call them medical geneticists here in London in the UK. And I have been studying a group of overgrowth intellectual disability syndromes for the last 2 decades. And so I'm going to tell you a little bit about my work with TBRS today. So in this talk, I would like to just first of all describe how we first identified DNMT3A variants as a cause of a new overgrowth intellectual disability syndrome. Then I'm going to think a little bit about what DNMT3A is, and then the different variant types that affect DNMT3A, before then we move on to the meat of this talk, which is really about the TBRS phenotype, or the clinical presentation. At the end, I'll touch on management guidelines and then think about the next steps. So as I'm sure all of you are aware and has already been alluded to this morning, we first identified DNMT3A variants as a cause of a new overgrowth intellectual disability syndrome, or an OGID syndrome, back in 2014. And we were very fortunate in being able to harness these new genomic technologies called whole exome sequencing at the time. That's now really been superseded by whole genome sequencing, to screen or analyze the DNA from a number of children who had been recruited to our study who had very similar phenotypes, so very similar clinical presentations, and shared the same characteristic facial features. And we identified 2 individuals in that study who had variants in this gene called DNMT3A. So we thought this was really interesting and extended our studies then to look at a further 456 individuals and identified a further 11 individuals with DNMT3A variants. So that brought that total to 13 individuals. And this is the paper that we published back in 2014. And in that paper, we pulled together some preliminary phenotype or clinical data. And we showed that all of those 13 individuals had a learning disability, and that they most frequently had a learning disability that was classified in the moderate range. All except 1 individual were classified as having overgrowth. And for the purposes of our study and in all our publications, we describe overgrowth as being height and or head circumference, at least 2 standard deviations above the mean. So that's some individuals just have height, some individuals just have a larger head circumference, and some individuals have both. And 2 standard deviations above the mean equates to the 98th centile on the WHO growth chart. So that's that second from top line on those charts. And all the individuals within our first cohort of 13 shared facial features that you'll see in the photo here. And we describe, in clinical genetics, we use all these fancy terms to describe facial features. And so when you read papers, You'll see phrases such as palpable fissures, which is the eyelids, the gap between the upper and lower eyelids. So we describe as narrow palpable fissures. And facially, the face is often round in shape, and then children and adults often have heavy, horizontal, quite low-set eyebrows. And so we pulled this together initially. We also noticed that many children had larger frontal incisors, and then when we looked at feet, we noticed that many individuals had shorter toes that were often widely spaced. And we were really interested in what DNMT3A was and what it does. So I'm just going to take a little bit of a backtrack, and many of you in the audience have a scientific knowledge, but I'm going to assume no scientific knowledge here at all and just pull us back to the sort of fundamentals of genomics. So DNMT3A is a gene that is located at chromosome location 2p23.3. So this is a gene. So this is one of my favourite pictures, which is the double helix. So the double helix is DNA, and a gene is a segment of that DNA. And I often describe to people that's a bit like a twisted ladder-like structure. And the rungs of those ladders are our bases. And C always goes with G, and A always goes with T. And it's alterations in those bases that we refer to as variants, and they were previously called mutations. And I'll come back to that a little bit further down the line. And when I'm describing genes and chromosomes to families, I often use the analogy that a gene is a bit like a book, and that when we're doing genetic testing, we're reading that book to see whether we can identify spelling mistakes within that book. And we have about 20,000 genes. So if you imagine a library with 20,000 books in it, and those books in that library are arranged into book stacks. So those book stacks are like your chromosomes. And so your chromosomes are made up of your genes. And we have 23 pairs of chromosomes, and they're labelled 1, the biggest, through to 22, the smallest, and then we have our sex chromosomes. So males usually have an X and a Y, and females 2 X's. And so this 2p23.3 is a bit like a map reference. So we're talking about chromosome 2, and you'll see that on chromosome 2, that is divided into a short and a long arm by a waist. And the short arm is called P for petit and petit, and Q is the long arm, and it's called Q just because it comes after P. And you'll see that on those chromosomes, there's these stripes, and we call those bands. And so that 23.3 really refers to which of those stripes that gene is sitting at. So it's really a reference as to where that gene is. And the protein that the gene DNMT3A encodes is an epigenetic regulator. And I'm sure that Peggy will talk a lot more about that in her talk and other of my colleagues later on. But really the epigenome is the orchestration of the genome, switching genes on and off. And if you think about it, you've got the same DNA in your eye as you have in your toe, but very different genes are expressed to make your toe the toe or your eye the eye. So the epigenome is that orchestration. It's switching genes on and off in the right place at the right time. But I'm sure my colleagues will talk a bit more about that later. And as has already been mentioned quite a few times, the TBRS Nord registry has been established to really start gathering more information about TBRS and DNMT3A. And I'm going to introduce this now before I come on to the next sections of my talk. This is an amazing initiative. It is an amazing repository of information. And it's really owned by you as a family organisation. It was started by you. It's managed by you. It is the most phenomenal method of driving forward our understanding of TBRS. It launched in 2021 in collaboration with Nord, and one of the great powers of this registry is that the data is entered by you and it's updated by you. And it's really talking to the point earlier, the importance of that natural history information, that longitudinal information. and really making sure that we got not just a snapshot of TBRS, but that ongoing, evolving understanding. And the data from that registry is available to the clinical and the scientific and research community on application, and I'm sure that Jill and colleagues can talk more about that if needed. So I'm going to present some data from the registry today, and that is data from a 23-month period that was collected between September 21 and August 23. Now, today I'm going to include snapshot information. We haven't yet got enough longitudinal information really to think about those time points, but that's going to be really critical going forward. So I'm going to include the most updated information where those families who have been brilliant and gone in and updated their information, but it's the most recent one that I'll present, from 19 different data collection categories, which equated into 19 different spreadsheets. And a big thanks to Kit, who did the most phenomenal job of making that data much easier for me to then analyse. And of course, huge thanks to all of you who have input your data into that registry and also updated it. And also a thanks to Eric, who did an amazing job. We had a bit of a chat about the DNMT3A variants, and one thing that we were talking about yesterday, and I think we really need to pick up on, is the inconsistency with which laboratories report DNMT3A variants. So Eric did a great job at, first of all, looking at variants of uncertain significance and reclassifying them in the context of our current knowledge and using what we call the ACMG framework, which is the framework that laboratories use in order to classify variants. And also ensuring that we had that consistency. So where a variant was reported as being likely pathogenic or pathogenic, there was consistency as to how that was reported. And I think that's a really big task for us, to ensure that there is that consistency between how laboratories are reporting on DNMT3A variants. So we had 111 data entries from patients and families. Unfortunately, the variant details weren't included for 7 individuals, so we had to exclude them. And then for a further 12 individuals, the variants were classified as of uncertain significance, so we also had to exclude them for the time being. I hope that there'll be reclassification of those variants and we'll be able to include them soon. So for the purposes of the information that I'm feeding back now, this was based upon 92 participants who had likely pathogenic or pathogenic variants in DNMT3A. So we would say that that is a causative DNMT3A variant, and therefore that is a diagnostic report. So I'm going to move into the third bit of my talk, having introduced the NORD registry, thinking about the different types of DNMT3A variants that you can have. So this is what we identified from the registry. So these are the 92 individuals, and you'll see that the predominant DNMT3A variant type was a missense variant. I'm going to describe what I mean by that in just a moment. And there are a number of missense variants that occurred in more than one individual, and there is a missense variant which is very common and that you will have heard talk about, called ARG882. And again, I will come back to that. The next commonest variant type was frameshift variants, followed by splice sites. We had 9 individuals with whole gene deletions, so missing the whole of DNMT3A, and then there were a few with nonsense variants and in-frame variants. So I've just thrown a whole load of words at you, so I'm going to just describe what those different variant types are. And the ones I'm going to really describe here are the missense variant, so that commonest variant type, a stop-gain variant, and a frameshift. And in order to help me describe this, I'm going to use this sentence: the cat was fat. And the reason I've used that sentence is because they're all 3-letter words. So if we just sort of go back a little bit, when I was showing you that slide with the double helix, and I was showing you the rungs of the ladder, you'll remember I talked about the different bases, the A, C, T, and G. So every 3 bases encodes for a single amino acid. We call that a codon. So 3 is important here. And so I chose those words on purpose. And the red box at the end is a stop. So the word fat means this is the end of the sentence. So a missense variant is when you get a substitution, so a letter change that changes the meaning. So the sentence structure stays the same, but now you see, rather than saying the cat was fat, we're saying the bat was fat. And so there's an alteration to a base there that changes that meaning of that word. And as I say, that is the commonest variant type in DNMT3A. It is also the commonest variant type in all of us. So if I were to sequence all of your genomes, I would identify about 5 million spelling mistakes in all of your genomes, and that's called background genetic variation. And the majority of that background genetic variation are these missense. They don't normally cause a protein change, but they're that background. And so one of the things that's really difficult for laboratory scientists is when they identify these variants, it's knowing whether that is causative or whether that's one of those background genetic variants. And that's why so many people have reports that say a variant is of uncertain significance, because a missense variant has been identified and it's unclear whether that's that normal background or not. So then a stop gain or a nonsense variant is the introduction of a premature stop codon. So you can see there, if C is now changed to an F, the sentence stops earlier. So that truncates that protein, it shortens it. And then finally, the frame shift variants are either a deletion of one or several bases or an introduction, a duplication, which shifts that frame. So remembering the importance of the 3-3-3, if you lose that structure, that sentence no longer makes sense. So that's a sort of very simplistic overview of these different variant types. But when you get those reports, these are the sorts of things that will be written on that report. And it's really explaining what is going on at that gene level. The splice site variants are to do with the bits that the chapters that are introduced or deleted from that protein. That's a bit more complicated to describe. And then the deletion is when you're missing the whole of the gene. So this is a schematic of the DNMT3A that I showed yesterday. And this shows those missense variants along the top with the gold triangles. And as I mentioned just before, there are a few missense variants that occur in more than one unrelated individual. And you'll see at the end in that methyl transferase domain, there's a variant at RJ2, and that's what we call a hotspot variant because it occurs time and again. We see many people with variants at that specific place. And then along the bottom, we have our frame shift variants. So you see there are more missense variants than there are frame shifts. The missense variants tend to target those critical domains with just a couple of individuals outside of those domains, whereas the frame shift variants occur throughout the gene. So let's now move on to what we know about the TBRS phenotype. And I think we're still at the beginning of our journey. We've made considerable progress in understanding the clinical features associated with TBRS, but there's lots more still to learn. And I think everything that's been put in place to learn through the registry, etc., is the way to improve our understanding. And it is just extraordinary, the progress that has been made already. So this is a schematic of our learning regarding phenotypes. So starting back in 2014, where we, as I mentioned earlier, described the initial 13 individuals. And then back in 2018, after we recruited individuals from around the world, we reported a paper with 55 individuals, really improving our understanding of the phenotype. And you'll remember from that initial slide, I was talking about the real cardinal features of TBRS being a learning disability, being overgrowth, and the characteristic facial appearance. In this 2018 study, we really began to expand beyond that and realised that kyphoscoliosis was common, hypermobility was common. But this was the first time that we really started realising that obesity was common as well in TBRS. And that led us to do an obesity study, and we had actually 72 individuals that were included in that study, and 74, sorry. And my colleague Sana Ulsters pulled together a poster, which is over there, really outlining the details of that study. So again, huge thanks to everyone who participated in that study. And it was an online survey where we really asked people about their weight at different ages and started thinking about eating behaviours. And in order to do that, we used a questionnaire called the Dykens questionnaire, which has been used many times in Prader-Willi syndrome and is a very validated way of investigating hyperphagic behaviour. So hyperphagia describes an increased desire to eat and not being able to stop eating as well. So it's really looking at different aspects of that behaviour, and that's detailed in Sana's poster. So through this survey, we were able to identify that 65% of individuals would be categorised as either being obese or having increased weight. And the onset of that was in early childhood, mostly at younger ages than 5, and that this was really being driven by this hyperphagic behaviour. So children really seeking out food, and then when eating, not really having an off switch, so would continue to eat. And we were talking yesterday about, and I think again Peggy might pick up this later, Peggy and Harrison have done a lot of work on mouse models, really thinking about the obesity phenotype in mouse models of TBRS. And really demonstrating that it's an interaction of different features which are really driving that obesity, the hyperphagic behaviour, but also larger adipocytes and really that sort of the endocrine control of the eating. But I'm sure that Peggy will talk to you a little bit about that later. So we now move on to the registry, and last year I presented some data on the first 62 individuals who'd completed their information, and now I'm going to update that with these 92 individuals that I mentioned earlier. And this figure was pulled together by my colleague Lily Barnett, who I think it's a very nice illustration of the various different body systems that are involved in TBRS. So those areas that I've boxed in red, I'm going to talk about in detail. So before I do talk about them, I just want to talk about the various different bits that are not in boxes. So some individuals did have brain MRI findings, which were mostly ventriculomegaly and Chiari malformations. Now, a really important thing that we were, again, talking about yesterday is that many individuals do not have brain scans. So we don't know whether some individuals have brain changes or not, because we haven't done an MRI scan. So within the UK, we would only give a child a general anaesthetic to do a brain MRI scan if they had a reason to do that. And that would be a change in cognition, that might be the new onset of seizures. There would need to be something that would then mean that general anaesthetic was justified. So the low incidence here may be the fact that actually we haven't done brain scans on many individuals. Over half of individuals wore glasses, mostly for reading. And as you are all, I'm sure, aware, teeth can be a problem. And I was particularly interested to hear about dentistry and the importance of dentistry and having access to dentistry, because this is so critical. So in TBRS, dental crowding is really common, and where a child has dental crowding, it can be much harder to maintain that oral hygiene. As I mentioned earlier, children and adults often have large frontal incisors, and then actually it was quite interesting that a number of individuals reported that the baby tooth were late to be lost. Renal disease was not common, and that's important when we're thinking about surveillance and management in TBRS. An umbilical hernia was reported quite commonly, as was constipation. So again, important to be aware of that. And hypertonia, which is floppiness, occurred in about 2/3 or nearly 2/3 of individuals. And that was usually in that newborn period, so soon after a baby was born, and tended to resolve as an individual grew older. Hirsutism, or increased hair, often on the lower back and the legs, was frequently described. And then pes planus, which is flat feet, and then talipes, which is the incurving of the feet, was also described. So if we come up the other side, 82% of individuals had hypermobility. So we would describe that as double jointedness or ligamentous laxity. And this is really important, and again something that came up in our discussion yesterday, because hypermobility can cause considerable joint pain in individuals. And it was a question that I asked because in some of the priorities, pain, especially in those teenage years, has been reported as a really significant feature. And I'm interested to know whether that might be related to hypermobility, because actually good, I was going to say physiotherapy, but in the States you'd say physical therapy, is really, really good way of mitigating that hypermobility, really building up that muscle strength around the joint. So really doing the job that the ligaments find it more difficult to do because they're stretchy. So it's really worth thinking about that and making sure that exercise, just walking is enough, but is maintained. And then eczema was reasonably common, and recurrent ear infections were reported in around 1/3 of individuals, with a hearing loss in 21%. Now, the recurrent ear infections often led to glue ear, for which grommets are often used. And I don't know whether that 21% of individuals were reported with a hearing loss, whether that was a conductive hearing loss, which is what you would tend to see with glue ear. So I suspect the two are related. And then obstructive sleep apnea was reported in nearly 20%. Again, really important to be aware of. So if your child is snoring, if you're worried about obstructive sleep apnea, it's really important to get a sleep study done to know about that. So I'm going to drill down into those clinical features that I boxed in red. So I'm going to start with growth. So 91% of the individuals in the registry would meet those criteria that I mentioned earlier for overgrowth. And so height and or head circumference at least 2 standard deviations above the mean. 60% of individuals had tall stature. 69% of individuals had a macrocephaly, which is describing a bigger head circumference. And on these graphs here, you can see that 2 standard deviation line is shown by the red dashed line. The mean is the 0. So you can see that actually pretty much everyone, the head circumference and height was above the mean. In the registry, 50% of individuals had a weight greater than 2 standard deviations. And then coming on to learning disabilities, everyone in the registry had a learning disability, and this was most frequently in the moderate range. So there's a couple of things just to mention from this graph. First of all, nearly 1/3 of individuals had an unclassified learning disability, so were described as having a learning disability, but that couldn't be categorized as mild, moderate, and severe. In part, that was because of the information that was included. I'm sure it's also because children were just too young to make that call. It's also really important to remember that at the moment, we tend to do genetic testing when a child or an adult presents to medical services, often with a learning disability. So that introduces a natural ascertainment bias. And again, we were talking a bit about this yesterday, because actually this becomes a self-fulfilling prophecy that if you're only testing individuals who have a learning disability, then of course the percentage of individuals will be 100%. And another condition that I do a lot of work on called Soto syndrome, and I feel as though a lot of people have heard me say this yesterday, but we are learning actually that about 10 to 15% of individuals do not have a learning disability. So I suspect this figure will change as there's increasing access to genomic testing, and so I suspect 100% isn't quite right. The other thing to say is that's a very subjective categorization. And when we start to use IQ scores, which are more objective, you'll see that there's a bit of an overlap between those individuals from the registry who are classified as having a moderate learning disability and those individuals that have a severe learning disability. So a more robust measurement of learning is an IQ score or a score of development. And so where possible, it's really, really important to include those data within the registry. Moving now to behavioural and psychiatric issues. And I think this is a little bit of an arbitrary separation, actually, because, for instance, anxiety, does that sit in behaviour? Does that sit in psychiatric issues? So I think almost these two should be merged. The key things from this is autistic spectrum disorder was very, very common and described in many, many individuals who participated in the registry. Anxiety is also very common. So those are the really two common things. There are a number of psychiatric issues that were not commonly reported, but were reported. So these percentages are the percentages of the 21%. So the 11% bipolar disorder equates to 2 individuals in the registry. So it's not a high number, but we are seeing psychiatric issues. And this is another reason why that longitudinal data, so those updates to the registry, are so important. And snapshots like this will only give us an idea. It doesn't give us the accurate information that is really, really important. And something that we have only recently realised since the launch, the establishment of the TBRS registry, is that cardiac anomalies are really common in TBRS. Now, that didn't come out from our original 2014 study. It didn't come out from the 2018 study where we had 55 individuals included. This has come from the registry. And nearly half of individuals with TBRS have been reported with some sort of cardiac anomaly. The most common is an atrial septal defect. So the heart has 2 chambers at the top and 2 chambers at the bottom. The atria at the top and the ventricles are the really big muscular bits at the bottom. And between those 2 atria and between the 2 ventricles is a septum. So an atrial septal defect is when there's a hole between those 2 atria. So that is the commonest heart anomaly that is identified in TBRS. That's closely followed by mitral valve prolapse, which is the valve that goes from the atrium into the ventricle. So that means rather than that being one way, that when the ventricle contracts, the valve goes back up into the atrium and the blood comes back. So that's mitral valve prolapse or regurgitation. And when we looked at the ages of those different cardiac anomalies, you can see that they are being diagnosed at the times when you would expect them to be diagnosed. But this was really important because this really determines the surveillance that we should be offering. So you can just about see that orange bar at the bottom, which is a PDA, which absolutely you'd expect to be diagnosing at birth. Then the septal defects in grey are in those very early months, followed by the valvular problems and then the conduction rhythm disturbances. And you can see that in that blue bar, that is occurring over many, it's a large age range. So it's really important to remember that because we need to be monitoring to make sure that conduction rhythm disturbances aren't occurring and responding where we think they might have done. And then this is just a graph thinking about the incidence of those different cardiac abnormalities, and in pink it's showing those individuals who have required surgery. So another important clinical feature we need to think about are seizures. So this is very specifically afebrile seizures. So that is seizures that do not occur in combination with a temperature. The seizures that occur where you have a temperature as well are referred to as febrile seizures. So, and febrile seizures are very common. And so when we think about seizure phenotypes, we tend to not include something that's very common when we're thinking about a very rare condition. So nearly a quarter of the participants in the registry were reported to have some type of afebrile seizure. The most common is a focal seizure, and you'll see the different types of seizures on this graph, but there were many individuals for whom the type of seizure was not reported. Importantly, most of these individuals were reported as having seizures. The diagnosis was when they were younger than 10 years old. There was just one individual with the temporal lobe epilepsy who was diagnosed at 40 years. So mostly, according to this preliminary data, and it is still preliminary data from the registry, you would expect the seizures to mostly have occurred in that younger childhood. Kyphoscoliosis, so a curvature of the back, is common. So kyphosis is a back-to-front curvature, and a scoliosis is a side-to-side curvature. So this is quite common. We often see kyphoscoliosis going hand in hand with hypermobility. This graph shows the age of diagnosis, again important when we're thinking about surveillance and ongoing management. So we had age for 25 of the registry participants. The youngest age of diagnosis was 1 year of age, and the oldest age of diagnosis was 36. But the majority of individuals were diagnosed when they were less than 15 years of age. And finally, really importantly, an area that we are really want to try to identify is what is the incidence of cancers and tumour types in TBRS. 8 participants in the registry were diagnosed with a type of cancer, and these are all detailed here. You can see as a range. AML was diagnosed in 2 individuals, one individual at the age of 12, another at the age of 19. And AML we're particularly looking at because we know that acquired variants, we call those somatic variants, so not variants that you were born with, but variants that you acquire during your lifetime. So somatic variants in DNMT3A can drive the development of AML. So we knew about that. So the question is whether AML might be associated with TBRS. And so there are 2 individuals in the registry, and then these are the reported cases. So these are published reports, and so a lot of work is being done to identify incidents, understand mechanisms. What I would say, and again, we were talking about this yesterday, the incidence doesn't seem to be high. There are certainly individuals who have been diagnosed with AML and different cancer types, but it's not more than 10%. It seems to be less than 10%, but there's a lot of work that needs to be done in this area, both in that, again, longitudinal data, making sure that we haven't just got a snapshot, and lots of work that's being done with scientific colleagues, really understanding DNMT3A and the functions and the interactions, and whether there might be a mechanistic overlap between TBRS and the somatic variants. And then finally, just wondering about time, finally, we have started thinking about genotype-phenotype associations. There's lots of different ways that we can look at this. So this is it's a preliminary, it's a rather crude analysis at the moment, where we were just looking at, does the type of variant relate to the severity of learning, the growth, the types of clinical features that an individual might have. And so the types of variants, we had 3 broad categories: the missense variants, the loss of function variants, or the deletions. And then these are the categories that we looked at. There's lots more work to be done here. We need much larger data sets to more robustly evaluate those associations. But more specifically, is there, rather than doing from variant type on an individual basis, does that equate to different individual variant types? And we were again talking yesterday about where those variants, which domain they targeted, and whether that also influenced the clinical presentation. So there's lots of work to be done here, and it's really important because if you can identify any associations, One, it's important for therapies and seeing response to therapies, but it's also important in terms of giving prognostic information and guiding management. So this is an area that would be really, really important to get more data on and be able to pull together a more robust evaluation and really start to tease out these patterns. There was only one thing that gave some signal from these analyses, and that was for hypermobility being more frequent in individuals with the frame shift nonsense types of mutations. Now, actually, it's not robust, that association. The numbers just aren't great enough to pull those out. So the final bit, I'm going to really think about management guidelines, and that's really distilling all of that information into what would we suggest your children as individuals. You are managed both at diagnosis and then at follow-up in that regular surveillance. This will be iterated, this will be updated and modified as we learn more, and that's a really important part of this. So we'd suggest that at diagnosis, a baseline echocardiogram, so that's a screen of the heart, is undertaken, and referral to physical therapy for hypermobility, and really to learn about joint health and building up that muscle strength around the joints. It's helpful to have a baseline hearing assessment and a baseline vision assessment. And then at that regular review, it's important to think about the behavioural issues, the psychiatric issues, really monitoring for kyphoscoliosis, asking about seizures. Dental issues are really important, and making sure that your children are under good dentists who are able to properly assess your children's your dental care. Sleep apnea is important to ask about. Constipation is common, and keeping an eye on that. And then again, hearing assessment. And then the ongoing cardiac follow-up with echocardiogram, and really thinking about the rhythm disturbances and conduction defects with an ECG. And then for the weight gain, thinking about practical and lifestyle interventions. And this is an area that I think it should be quite accessible in terms of pharmaceutical agents that we might use, and discussions are being had in that space, because that, I think, when we're thinking about management, and particularly pharmaceutical management for TBRS, I think this is one of the first areas that we may be able to think about targeting. And then the big question is, should we be doing any tumour screening in TBRS? And particularly, should we be screening for AML? And people are really thinking about that now, and we need to learn again, understanding the incidence, and then thinking about, if we were to do screening, what would that look like? What I do say to families that I meet in terms of that is to really have a low threshold for seeking medical investigation if you are worried. And so really thinking about if your child is having recurrent infections when they didn't have beforehand, if they're particularly tired when previously they wouldn't have been, if they're bruising easily, if they're having lots of nosebleeds, things that you have noticed as parents, and you know your children. If you've noticed a difference, then ask the question and speak to someone, and then they can guide you as to whether other investigations are needed. So this meeting, which I have to say is the most brilliant meeting, is really thinking about next steps and harnessing the engagement, the skills from the research and the scientific and the clinical community, and bringing everyone together and to think about how we can start driving forward and moving our understanding of TBRS and realising that ultimate goal of full understanding of the TBRS clinical presentation, and then thinking about management and therapies for TBRS. And the journey since 2014 has been extraordinary. I work with many different family groups. As I said earlier, I work on this group of conditions that we refer to as a family called OGID syndromes. This group, the advances that your group has made is extraordinary, and it's the deepest pleasure to work with you all. It is absolutely phenomenal. And since 2014, we have learnt so much more about the clinical presentation. It remains that the cardinal features are a learning disability, the characteristic facial appearance, and overgrowth. But we now know that very commonly associated are hypermobility, kyphoscoliosis, obesity, and cardiovascular disease. The current management guidelines is based upon the current knowledge and the age of onset of those different clinical features, but that will iterate, that will be updated, that will need modification as we're learning more and as we get more natural history data, as we learn about genotype-phenotype associations. So we are very much at the beginning of this journey, but we're going along this path quite quickly. And as I said, it's only through collaborative networks like this, everyone working together, that really drives forward our understanding. So there are many, many people that I owe huge thanks to, and funding organisations, but none more so than this group. And I was saying yesterday with Jill that this is a photo from 2019, and we absolutely, one of my missions of this summer, to get a new photo, because it's with huge thanks to all of you that working with you is such a deep pleasure. So thank you very much for embracing me in your community.